Hey everyone, welcome back. So in our last video, we wrote a custom Python script to scan a FASTQ file and manually chop off the low quality ends of our DNA reads. That was a fantastic coding exercise, but here is the truth. When you are working with a real data set containing like millions, like 15 millions reads, you don't write a custom Python loop to check the quality normally. Okay, you use the industry standard. So today we are heading back into the Linux terminal to learn FastQC. It is a tool written in Java that instantly generates a beautiful comprehensive HTML report of your sequencing quality. Okay, so if you do NGS analysis, you will use FastQC basically every single day of your career. So yeah, let's get started. This might be something that people who are already in biology or genomics might know, but if you are shifting from, you are someone from computer science background, this might be good exercise for you. Okay, so yeah, let's open up our terminal in VS Code over here. And if you're on Linux or using Windows subsystem for Linux, installing FastQC is incredibly easy. We can use the standard package manager. So yeah, let's start WSL first of all. Then after that, once WSL is started, you can run sudo apt get update. Okay, it will ask you to enter your password, and once that is done, okay, after the update, let us do sudo apt get install FastQC. So FastQC is already installed on my system, but if you run this command, it will install FastQC for you. But as you can see, FastQC already exists over here, so basically nothing happened. Okay, and if you're using Conda, which is very common in bioinformatics, you can simply type this command over here, Conda install C bioconda FastQC. Okay, once it is installed, we can verify it is working by checking the version. So yeah, let's check that FastQC version, and yes, FastQC has been installed. Okay, now let's run it. I have downloaded a small FastQ file, not exactly small, but it has quite a bit of reads over here. Okay, so yeah, so this file, we will be running FastQC on this. But it will take exactly one line of code over here. Okay, so let's run that FastQC. Then we will enter the name of the file, sample reads.fastq. Okay, and as soon as you hit enter, you will see this text flashing on the screen as FastQC processes the files. It is analyzing the GC content, the sequence length, and the Fred quality scores for millions of bases in a matter of seconds. And when it is done, you can basically type ls to see what it has created. But since already some files are there, so I will just point it out for you. The two new files that we got are this one HTML file and this one compressed file, right? So this HTML file is what we want. Okay, so let's try to open that in a browser. I'll show it to you how it looks. Okay, okay, so since it is an HTML file, you don't open it in the terminal or in a text editor. You open it in your web browser like Chrome or Firefox. In VS Code, you can usually just hit the right click the HTML file in your Explorer panel and select open in default browser. I have opened it in Edge for your understanding, right? So when you open it, you are greeted by the FastQC dashboard. On the left side, there is a summary menu with a traffic light system, right? And a green check mark means that module is great. Okay. Yellow exclamation mark is a warning, and the red X means the data failed that specific quality metric. Okay. So, but don't panic. If you see a red X, FastQC is very strict basically. So, and sometimes biological realities like sequencing highly repetitive RNA will naturally trigger a warning. So, you have to interpret it with biological context. Okay, so let's look at the basically the most important graph in all of bioinformatics, which is the per base sequence quality plot. Okay, not this one, but this one. Okay, so yeah, on the x-axis you have the position in the reads in base pairs from base 1 then all the way to the end. Okay, and on the y-axis you have our old friend, which is the Fred quality scores ranging from 0 to somewhere around here. Okay, The background is painted in three colors basically. The green, which means very good quality, orange means reasonable quality, and red means poor quality, or which also means very high error rates. Okay, so for every position across your millions of reads, FastQC draws a yellow box and whisker plot. Okay, so yeah, the red line in the middle of the yellow box you can see over here is the median quality score for that specific base position. So in a gray data set, all the yellow boxes will be floating high up in the green zone over here across the entire graph. But remember what we learned about Illumina sequencing, right? The chemistry degrades over time. So it is perfectly normal to see the yellow boxes slowly dip down into the orange or red zone as you get to the right side of the graph, which is the end of the reads. But in this file, you can see basically everything is in the red zone. So this is a very bad quality read. Okay, so let's quickly look at the two other essential tables, tabs on the left. First is the per sequence GC content. This shows a bell curve of the GC percentage across all your reads. It also overlays a blue line showing the theoretical distribution. Okay, if your red line has weird jagged spikes or two peaks or something like that instead of one, it usually means your sample is contaminated with another organism's DNA and you have adapter contamination. Okay, second, let's check the adapter content. Uh, at the very bottom, you can see adapters are artificial pieces of DNA glued to your sequences so the machine can read them. So if they accidentally got sequenced along with your actual biological DNA, this graph will shoot up. And if it does, you absolutely must trim them off before aligning your data. Okay, and this is FastQC basically. You can check out the other tabs also after you run your own tests. So you now know how to generate an industry standard quality report from the command line and how to read the most important diagnostic plot in genomics. Okay, so what do we do if our this FastQC report shows a big red X for quality at the end of our reads? Okay, and a huge spike in adapter content as well. So what we have to do is we have to clean it up. We cannot use our simple Python script for adapters. We need a professional command line trimmer. Okay, well, Python also supports that, but the industry standard is using a command line tool. Okay, so in our next video, we are going to learn how to use those tools to automatically slice off adapters and draw low quality bases using the terminal. Drop the low quality bases. Okay, so try downloading a raw FASTQ file from the SRA database and running FastQC on it yourself. Okay, if this video helped you visualize your data, please hit the like and subscribe button. I will see you in the next video.